So, good afternoon again, and in the series of lectures of Fundamentals of Entomology 1, after finishing the important factors and also after finishing various systems, today we are going to discuss about the sense organs in the insects, and we may have a doubt in most of the occasions that how these insects will have a smell, can sense the smell, how these insects can sense the taste, touch, how they can see various objects, what are all the ears as far as the insects are concerned. Yes, like on the left side picture with the cat, similar to the other organisms and also human beings, the insects are extremely developed as far as sensing various stimuli. They are in fact better. They are in fact better compared to the humans. So this class we will try to understand what are all the organs present. Basically, as far as the insects are concerned, we can classify them into 5 major categories. The organs of vision, the organs which are helpful for seeing, for visualizing. They are nothing but photoreceptors. Then the organs of hearing, just like ears, as far as humans are concerned, other higher organisms are concerned, that we call them as auditory organs. Then insects also have a wonderful and very, very highly developed organs of smell. They are chemoreceptors, and they have organs of touch, tactile receptors. They have organs of taste as well, that we call them as gustatory receptors. So it means the insects, the beautiful 6-legged, 4-winged, segmented body, wonderful insects. So they have various organs to sense the vision, touch, smell, to taste the food, and also to hear the sounds. The first and most important thing we will be discussing about the visions of organ. The visions of organ we have actually discussed in the second, third class itself that they are highly successful because they have compound eyes. They have many number of eyes, the smaller eyes within the big eye. So even if one smaller eye is damaged, absolutely no problem as far as the vision is concerned, because they have many, many number of eyes within that compound eye. So that is one of the most important organs of vision as far as the insects are concerned. Besides the compound eyes, they do have some smaller eyes that we'll be discussing a little later. The compound eyes, just look at cockroach at your home. Just try to look at the compound eyes of a grasshopper which is roaming nearby home. And try to look at the compound eyes of the bigger ant or the honeybee or the moth which is coming to your home. The compound eyes are present in all the insect species except in Protura and Diplora. It means they are blind. The Protura and Diplora are aterygota insects. They are primitive insects. So the compound eyes are present in all the adult forms, whether it is a hemimetabola or holometabola, and also in all the nymphal forms or immature forms or hemimetabola. But the compound eyes are absent in immatures of holometabola, which is nothing but larva or caterpillar. Remember, it means the larvae which you find nearby the weeds, nearby your home, they are blind. But they have a simple stemmata, so which is useful for the vision. And it is very, very simple. If the insect is parasitic, or especially the endoparasite, if it is living inside a body of another organism, it's very simple as like that. It doesn't require any compound eyes because it is actually parasitic. It means the compound eyes are present in all the insect except in the Aterogota, Procura, and Diplura, and also the larval forms or immature forms of the holometabola insects or the Endotergota insects. And also the compound eyes are reduced or absent in all parasitic insects. And look into this picture. Look into this picture. On the left side, they have a wonderful brown colored or reddish brown color compound eyes, and these compound eyes, they are actually almost closer to each other. Whereas look into the grasshopper here, the compound eyes, they are totally separated from each other. See here. So if they are totally separated from each other, this kind of compound eyes arrangement is called dicoptic. Di means two. Two are separate. They are separated. They are totally separated from each other. So that type of arrangement of compound eyes is called dicoptic. If the compound eyes comes together and closer to each one is called holoptic eyes, which you can see in this slide. So here the compound eyes coming very, very close. Absolutely, you can't differentiate between one to another except this small group. In some of the insects, so in general, in most of the insects, it is dicoptic. Almost like most of the insects, it is dicoptic, which means the compound eyes are separated from one to another. In very few, mostly in Diptera and some very few insects, it is holoptic, in which you can see the compound eyes are coming very closer. In both the sexes, whether it's a male or female, it is the same. But in case of tabanid flies, it's something interesting. But in case of tabanid flies, they are flies actually. In males, the compound eyes are coming very closer, which is called holoptic. In the female insects, the compound eyes are separate, which is called dicoptic. So you can easily identify which is male tabanid fly, which is female tabanid fly. That's called sexual dimorphism. That's what we have discussed. If you remember, even in case of antenna, in almost all the males, the antenna is very, very bigger because the male insect has to perceive the pheromones released by the female insects. So antenna is bigger in the male insects because it has to take all the chemicals, it has to absorb all the chemicals. So similar kind of sexual dimorphism, it means the males will have a holoptic eyes, the females will have a dicoptic eyes as far as the tabanid flies. So it means you can classify the compound eyes based on the arrangement. How closer they are, how distance they are. So if they are separated, the compound eyes are separated just like grasshopper you see, just like cockroach you see, it's called dichoptic eye. If the compound eyes are very, very closer from each other, it is called holoptic eyes. Yes, you go to closer to the compound eyes. So if you look into the closer to the compound eyes will have a number of smaller eyes. The compound eyes will have a number of smaller eyes or smaller units known as ommatidia. So it means the compound eye is an aggregation of ommatidia. The compound eyes is an aggregation of ommatidia. Now ommatidia numbers may vary from insect to insect. The number of ommatidia may be very, very high in number in some, maybe very, very few in number in some. The ommatidia is the unit of the compound eyes, and the number of ommatidia will vary from insect to insect. It is as simple as if the number of ommatidia are more, for example, you take the grasshopper and take a lens and go to the closer to the ommatidia, then you will see how they are arranged. If the number of ommatidia are more, they are usually arranged in hexagonal arrangement, just to accommodate more number of ommatidia. If the number of ommatidia are less, it is arranged in a circular shape. You can see on the left side picture here. So if the number of ommatidia are less, they will be arranged in circular arrangement. If the number of ommatidia are more, they are always arranged in hexagonal shape. It is as simple as like this. Now let us try to go into the structure of the ommatidia, how it looks like. A small eye, a unit in the compound eye. A typical ommatidia consists two things. One is optical part and the second one is sensory part. Similar to the humans. Like for example, we have an optical part and on the back side of the eye, you have a sensory part. Similar to you and me, the insects are also having the optical part and also sensory part. The optical part will receive the stimuli, will actually try to absorb the light. A light gathering apparatus. The optical part is a light gathering apparatus, and the gathered light will be sent to the nervous system for sensing. So ommatidia will have two parts. One is optical part and the second one is sensory part. The optical part we call them as a dioptric apparatus. The sensory part is actually a connection to the nerve cells. It's a nerve cell. So similar to the humans, they also have the optical part, and on the back side of the optical part is a sensory part. So let us try to understand what is the structure of ommatidia. We will see the optical part here. So the optical part, the red line, I just made a red line here to make you understand. This is all optical part. It is a light gathering part. This is the part which actually perceives the light gathering part. And this optical part, a light gathering part, will have two units inside. One is the cuticular lens and the second one is crystalline cone. So look into the structure of cuticular lens here. The lens, the optical part. The lens is the outermost transparent, colorless layer. You may ask a question. Sir, you say it is a colorless, but actually in the previous picture, I have seen it is a reddish brown in color in one fly, and it is in the grasshopper, it is green in color. That's different. That I will come back. I am talking about the lens, the outermost part. The outermost part is the colorless layer, is the cornea, which is acting as a lens, which is always a biconvex. You can see here, which is always a biconvex. And this cornea is secreted by the epidermal cells. The cornea is secreted by the epidermal of what? Epidermal cells of the exoskeleton. So the epidermal cells are nothing but corneagen cell here. They have two corneagen cells. These two corneagen cells will produce the corneal lens, which later, after they produce the corneal lens, their duty is over. So the corneagen cells will become the primary pigment cells after secretion of the cornea, because their job is over. So they will just go to the side and they become a primary pigment cells. So this is the first part as far as the optical part is concerned. Then we will try to understand the crystalline cone. So this is the cornea. The cornea is actually produced by the epidermal cells, which are nothing but corneagen cells. And these corneagen cells, after production of the lens, they become the primary pigment cells. So actually they will not be here, they will simply drag to the side and they become the primary pigment cells. And the second part is crystalline cone. You can see here the crystalline cone, the white color, the crystalline cone. The crystalline cone is actually a four cells, which is produced by a four cells, which are nothing but Semper cells. You can see here, this is the crystalline cone, and the crystalline cone is surrounded with the cone cells here. And these cone cells are nothing but Semper cells, and these cone cells will produce the crystalline cone. And this is called as a secondary lens. The primary lens is the cornea. The primary lens is the cornea, and the secondary lens is the crystalline cone. And this crystalline cone is actually produced by the cone cell, which is nothing but the Semper cells. There are actually four cone cells. Remember, the cornea is synthesized by the corneagen cells, two cells. Whereas the crystalline cone is produced by the four cells, which are nothing but cone cells. The eyes of the insects are also can be classified. Not necessarily all the insects will have a crystalline cone. In some, the crystalline cone, the secondary lens may be absent. So, the eyes in which the crystalline cone is present are called eucone, the real cone eyes. Eyes in which the crystalline cone is absent, we call them as exocone eyes. Now let us move into the receptive part, which is nothing but sensory part. The sensory part, it's as simple as like this. When you call it as a sensory part, it should be connected to the nerve cell or nervous system. Because the cornea takes the light and it goes to the crystalline cone, And the message, the light which is transmitted should be going to the nervous system as a stimuli. The sensory part, the most important part of the sensory part is the reticular cells. Reticular cells are nothing but the nerve cells. They are the very much elongated nerve cells. They are the receptive parts of the insect eye. Come to the top to bottom. When we discuss about the corneal lens, the corneal lens is synthesized by two corneogen cells. The next part is crystalline cone, the secondary lens. The crystalline cone is actually synthesized by the four cone cells, Semper cells. And the next part is the sensory part, and the sensory part is nothing but reticular cells, and the reticular cells are usually in 8 in number. So there are 8 reticular cells. So arranged in a circular manner. So there is a cornea, there is a rhabdome, and then followed by the reticular cells, which is actually a sensory part. And this reticular cells is a mixture of 8 cells and 8 nerve cells. So when 8 nerve cells are making some kind of circular arrangement, there is a gap in between, in the middle of the core. So the central core space is with full of microvilli, which is an extension of the nerve cells. And all these microvilli, very, very small extensions of these nerve cells, which are going to the core space, which become a rhabdome. And all these projections, we call them as a rhabdomeres. And all those rhabdomeres collectively form like some kind of rod. Like, just imagine, there is a rod in the middle of this sensory part, and that rod is surrounded by 8 reticular cells. Actually, that rod kind of part is because of the cellular extensions of the nerve reticular cells. And these extensions, we call them as a microvilli. All these microvilli together, they form like a rhabdome, which is basically, that is the part which actually sends the message. So again, similar to the optical part, because once a hematodia receives the light, and that light should pass it through only that hematodia, it should not divert to the next one. That's how there are primary pigment cells which actually avoid going of the light from one hematodia to another hematodia. And similarly, there should be some kind of insulation mechanism even for the nerve cells. So even for the nerve cells, there should be some insulation mechanism, which means the message received by these 8 nerve cells from one hematodia should go to the directly to the nervous system. It should not be distracted. That's how this secondary part, which is nothing but the sensory part, which is nothing but a mixed total of reticular cells, which is surrounded by a secondary pigment cells. The duty of the primary pigment cells and also secondary pigment cells, which are surrounding the crystalline optical part and also which are surrounded by the sensory part, is not to allow the message to go out of this hematodia. And now, as far as the reticular cells is concerned, like I told you, there are 8 reticular cells. And these 8 reticular cells, a lot of microvilli which is coming, cellular extensions which is coming into the middle core. And all these cellular extensions together, they form as a rhabdome. And these reticular cells, how they will send the message? They have a light absorbing pigment called rhodopsin. They have a light absorbing pigment. These reticular cells, they have a light absorbing chemical, a pigment called rhodopsin. Because the rhodopsin absorbs the light, it's a chemical, and that's how the message starts. The mechanism of nerve transmission will start from there. Something beautiful. So once the light is absorbed by the cornea, then the light will be sent to the crystalline cone, and the same stimuli will be going to the reticular cells where the rhodopsin is present, and that rhodopsin, the chemical, which pass on the message to the chemical procedure and finally into the brain in the form of electrical impulses. The chemical impulse will be converted into the electrical impulse. That's how the nerve impulse will go to the brain. That's how the insects will identify the object. Let us see how the image is formulated. How the image is formulated. There are many number of ommatidia in every insect. Umpteen number of, as I told you, sometimes it may be going to the 10,000, then 10,000 ommatidia, they receive the stimuli and how this image is formed. The image formation is varying because of two major differences. The major difference here is based on the gap between crystalline cone and the rhabdome. In the previous picture, I will go back to the previous picture here. You see here, the rhabdome is immediately attached to the reticular cells. So in this picture, there is no gap at all. This is the typical rhabdome. This is the typical ommatidia structure. So in this picture, there is no gap between the rhabdome and the reticular cells. For example, there is a gap between the crystalline cone and the rhabdome. The vision formation is different. So if there is no gap, all these ommatidia like previous picture, there are number of ommatidia arranged serially like this. When there is no gap between crystalline cone and the rhabdome here and the rhabdome, the vision is different. So when there is a gap between crystalline cone and rhabdome, the vision, the image formation is different. The eyes in which there is no gap between crystalline cone and the rhabdome is called apposition eyes. Apposition eyes. And these apposition eyes are always also termed as a photopic eyes. It means, so this kind of arrangement where there is no gap between crystalline cone and the rhabdome, and that kind of arrangement is always there in the day active insects. Because they pursue huge supply, they have to identify, they are very active. There should not be any gap, frankly speaking. If there is a gap, the message will not go directly. It can go to the next rhabdome. So the message received from the cornea, the message received from one crystalline cone can also go to the next rhabdome. In all these day active diurnal insects, apposition eyes are present, that we call them as a photopic eyes, because it is adapted for the bright light conditions. The eyes in which there is a clear gap between crystalline cone and rhabdome is called superposition, because the image is superimposed. The image received, the message received by one cornea will go to the next rhabdome. That's how it is called superposition eyes. And these superposition eyes, which always present in the night active, are nocturnal insects. and they are all adapted for the dim light conditions. Because there is a clear gap, between the crystalline cone and the rhabdome, these eyes are also called as a clear zone eyes. These eyes are also called as a clear zone eyes. In the apposition eyes, I told you that the light received by one rhabdome, one eye, one ommatidia, one lens, which goes to the same crystalline cone, then followed by the same rhabdome. And it will not go to the next rhabdome. It will go to the same rhabdome. So it means the light received from one facet passes through the same central portion and forms the image on the same rhabdome. Because there is no gap between the cone and the rhabdome. Besides the compound eyes, insects are also have a simple eyes. Besides the compound eyes, insects are also having the simple eyes. And these simple eyes are nothing but ocelli. You can clearly look into the grasshopper. Whenever you catch hold of the grasshopper, you just find there are 3 spots on the head. So these 3 spots are nothing but simple eyes. These 3 spots are always arranged in between 2 compound eyes. There are usually 3 ocelli, and these 3 ocelli are not very highly developed optically and sensory wise. They are only sensitive to very, very low light levels, and they don't have a bigger role. And these ocelli, they are highly reduced in case of cockroaches. They don't have a bigger role as far as the light stimuli perception is concerned, but however, these highly reduced ocelli are called as a fenestrae in case of cockroaches. As I told you in the beginning, that the caterpillars and the larvae, the immatures of holometabolous insects are the endopterygores. They don't have a compound eyes. They are blind one way. But however, they have these lateral ocelli, and which are the only eyes as far as these larvae are concerned. So lateral ocelli are the simple eyes which are present in between the compound eyes and the head. And these ocelli are not actually sensitive, very much sensitive. But however, these 3 ocelli which are present on the head, they are very, very important in some cases, like holometabolous larvae. Now let us move into the organs of hearing. Yes, insects do hear noises. And insects also make noises, you know very well. When insects make noises, there is a purpose. It means some other insect should hear or sense it. Insects are also having auditory organs just like our ears. They are the organs of hearing. Let us see what kind of organs of hearings are present in case of insects. There are 4 types of organs of hearing. there are four kinds of organs which are present in insects. But the most important organ, we call them as a tympanum. The tympanum is like a drum-like structure, which is composed of a thin cuticular membrane outside. And the thin cuticular membrane, and there is a nerve connection inside so that it can actually sense the sound. So usually this tympanal organ is having a thin cuticular membrane, and below the thin cuticular membrane there is some kind of nerve connection. And two insects are extremely popular as far as these auditory organs are concerned. One is the grasshopper and crickets, grasshoppers, and the second one is crickets. And in the grasshopper, always on the first abdominal segment, there is a tympanum. And as far as the crickets are concerned, it is always on the front legs. This is the tympanal membrane, which you can see, it's a cuticular membrane. Very thin cuticular membrane. So these tympanal organs are also a taxonomic feature as far as these grasshoppers and crickets are concerned. And the second organ is Johnston organ. If you remember, when we had a discussion about the structure of the antenna, the second segment, the scape, pedicel, and flagellum, the second segment, pedicel, will have an auditor organ, a sensory organ, which is nothing but Johnston organ. So the Johnston organ is nothing but an auditory organ, which is always present on the second segment of antenna on all the insects. All the insects. But it is extremely important as far as the male mosquito is concerned, which is used for the mating because the male detects the wing beat frequency of the female and is attracted to the female insects. At the time of mating, the female insects will have a separate sound making mechanism through wing beating, and that will be perceived with the male insects. How? Because of the Johnston organ. It means the Johnston organ, the auditory organ in the male mosquito is highly developed. And the third is auditory hairs, and they are present here and there, not very, very specific. The very, very micro auditory hairs, they are not extremely developed, but some auditory hairs are present in Lepidoptera and larvae. And the fourth auditory organ is the pilifer, which is present in the head of the sphinx moth at death's head moth. Basically, this is to perceive the sound produced by the bats. Bats are the enemies of these hawk moths. That is the reason they have developed a wonderful auditory organ called pilifer, so that the hawk moths or the death's head moths can easily hear the noises made by these insectivorous bats. So there are four auditory organs as far as the insects are concerned. One is tympanum, and the second one is Johnston organ, and some auditory hairs, and also pilifer. Now let us go to the organs of smell. Chemoreceptors. Yes, the organs of smell. The most important is the antenna. Why actually they should smell it? Why? What is the purpose? Extremely important for detecting the smell. Smell is nothing but a chemical, a chemoreceptor. order, a chemical constituent which is coming out of the plants, which is coming out of the enemies, which is coming out of the other sexes for mating purpose. So it needs to smell it. The smelling is the principal organ which smells. In the insect is antenna. The olfaction. All these volatile chemicals which are produced by the plants, which are produced by the other animals, which are produced by the other insects of the same species or other sexes, which is useful for finding the food, for finding the enemy, for finding the place for oviposition, for finding the place for living, and also for finding the mate. This olfaction, the sensing the smell, which is nothing but chemicals, the volatile chemicals, that sensing is done by the antenna. And the next one is organs of touch. Yes, they do have some structures all over the body, which can actually sense the touch. And also organs of taste, gustatory receptors. So as far as the insects are concerned, they are very, very sensitive as far as the touch is concerned, the tactile receptors. They have a huge number of tactile receptors across the body. The tactile receptors which can actually sense the touch. They are present on the antenna, they are present on the wing, they are present on the component, they are present everywhere. But as far as the sense of taste or gustatory receptor are concerned, these receptors are present in almost all the parts of the mouth parts. And you don't believe, even there are some insects, they can distinguish between sweet and sour, salty and bitter tastes. Such kind of evolution process is there in as far as the insect senses are concerned. And besides these 5 important senses, insects also can identify the changes in the humidity. Insects also can identify the changes in the humidity because of some sensilla, and most of the hygroreceptors are present on the antenna. Insects are also can sense the changes in the atmosphere pressure. Insects also can sense the heat, and that is extremely useful as far as the female mosquitoes are concerned. The female glossina can easily identify the temperature of the human before it inserts the stylates for sucking the blood. And the desert locusts, they can even identify the center of gravity, such a wonderful, the proprioceptors. So in the insects, these kind of sensory organs are present. And in this class, we try to describe and discuss about various sense of smell, sense of vision, sense of taste, sense of touch, and sense of taste. And the most importantly, as far as the structure of the compound eye and the structure of the ommatidia, which is inside the compound eye, which is a basic unit of the compound eye, and different types of ommatidia, different types of compound eyes, and how the vision is formulated, And also we try to discuss about the stemmata, then we try to discuss about various auditory organs, various other sense organs as well. So in this class, now we understand that the insects as brilliant as humans. They're extremely developed for sensing the light, for sensing the volatile compounds, for sensing the taste, for sensing the touch. And especially as far as the sense of touch is concerned, they have a huge number of sensilla spread over across the body. That's how these